Good morning, everyone. I would just like to thank the American Society of Mammalogists for this award and the opportunity to present some of my thesis research with you all today. I would also like to thank my co-authors, Dr. Lisa Shipley, Dr. Jennifer Forbey, and Dr. Janet Rachlow. Today we'll be sharing some of the research I've done concerning specialist and generalist rabbits and how they trade off plant fiber and plant toxins when consuming a shared food resource, sagebrush. Well, first you may be asking, why the sagebrush-steppe? Sagebrush-steppe habitats support many plant and wildlife species and is one of the most imperiled plant communities in North America. Alterations such as conversion to agriculture, intensive cattle grazing, and invasion of non-native plant species are influencing the suitability of habitat for a variety of wildlife species, many of whom are becoming species of conservation concern. Sagebrush has been shown to vary in dietary quality across spatial scales from landscapes to individual patches and shrubs to even the different parts of the sagebrush plant. For example, sagebrush leaves contain relatively high levels of digestible energy and protein year-round, but also contain high levels of plant secondary metabolites, which I will refer to from now on as PSMs or plant toxins, and especially the group of plant toxins called monoterpenes. Sagebrush stems, on the other hand, are likely to contain high levels of fiber but lower levels of monoterpenes. Therefore, sagebrush specialists and generalist herbivores may make different trade-offs between plant fiber and plant toxins when choosing which parts of the sagebrush plant to consume. Because sagebrush contains high levels of monoterpenes, most herbivores living in the sagebrush-steppe are generalists that consume relatively low amounts of sagebrush. These animals lack mechanisms for detoxifying high amounts of monoterpenes, and therefore they need to consume a variety of types of food to prevent from overloading one specific detoxification pathway. The few sagebrush specialists, on the other hand, have developed specific behavioral and physiological adaptations for coping with monoterpenes in their food, such as smaller body sizes, extensive chewing, and selection of plants with lower monoterpene concentrations or plants with a wider variety of monoterpenes. Therefore, we wanted to compare the behavioral adaptations to diet risks of a specialist and generalist rabbit species that occur sympatrically in the sagebrush-steppe to diet risks in this sagebrush. The pygmy rabbit, our specialist herbivore, weighs on average 1 pound and specializes on sagebrush, consuming up to 99% of it in the winter. On the other hand, the mountain cottontail is our herbivore specialist, or our herbivore generalist, and they weigh between 2 and 3 pounds and are dietary and habitat specialists, occurring in a wide variety of habitats and consuming a wide variety of food resources. In this study, we wanted to compare the use of sagebrush as a winter food resource using both field observations and controlled feeding experiments. In both feeding observations and controlled experiments, we expected the pygmy rabbits to consume a greater amount of sagebrush, but also a greater proportion of leaves than the cottontails because they are able to detoxify these monoterpenes to a greater extent. However, because we expected sagebrush to be leafier at the tips of the plant, and because cottontails are larger and have a longer retention time of food in their gut, we expected cottontails to crop the sagebrush branches at larger diameters and therefore consume more fiber. To compare the amount of sagebrush and the proportion of leaves consumed by free-ranging rabbits, we collected 10 fresh fecal groups from each species at 3 different field sites in Idaho, which were Cedar Gulch, Rocky Canyon, and Magic Reservoir. Feces were collected from one pile or one rabbit trail and were only collected if they were on top of the snow to make sure they were fresh at the time of collection. Feces were also collected if they were 20 or more meters apart to make sure we got a representative sample of the entire study site. We performed microhistological analyses on the feces at the Wildlife Habitat Lab at Washington State University to examine the percentage of sagebrush consumed and the proportion of leaves consumed by each species. We used two-way ANOVAs to compare the percentage of sagebrush and the proportion of leaves by site and species. To examine the selection of sagebrush diameters cropped by free-ranging rabbits at only the Cedar Gulch field site, we first identified browsing sites and confirmed species by 45-degree crop marks on the branches of the sagebrush shrubs, rabbit tracks, fecal pellets, and in the case of pygmy rabbits, burrow systems, and or a visual confirmation of either rabbit species. When species of browser was confirmed, we measured individual crop marks on a single sagebrush shrub on multiple branches on that shrub. We also wanted to determine if there was a relationship between the size of diameter cropped and the amount of leaves and stems that were present on that branch. To examine the preference pygmy rabbits and mountain cottontails showed for different parts of the sagebrush in controlled feeding experiments, we offered a bowl of each stems and leaves of Wyoming big sagebrush simultaneously to 8 pygmy rabbits and 5 mountain cottontails with either 50% or 25% of a normal intake of their pelleted diet. We also measured the NDF, or neutral detergent fiber, and the total monoterpene content of each the sagebrush leaves and stems separately. We used a two-way ANOVA to compare the proportion of sagebrush consumed, the proportion of leaves consumed, and the total dry matter consumed between rabbit species and level of basal diet. To determine the stem diameter of the sagebrush cropped by captive rabbits, we offered several sagebrush branches to each of 11 pygmy rabbits and 8 mountain cottontails during a feeding time during the night when we determined was their peak feeding time. All the crop marks made by each rabbit were identified and measured, and we used a two-way ANOVA to compare the stem diameters cropped between species. We determined that sagebrush leaves contain about 6 times higher monoterpene content than the stems, but about half the fiber content. We also found an increasing relationship between the stem diameter and both stem and leaf mass. So first, I would just like to explain this graph. On the x-axis, we have our field sites, and on the top graph, we have the pygmy rabbit diets from the free-ranging rabbit fecal samples that we collected, and on the bottom is the cottontail diets. So if you can't read the little key at the top I have, the white portion on the graph is the sagebrush consumed by each species. We found that pygmy rabbits consumed a significantly greater amount of sagebrush at all field sites, and both species consumed a greater amount of sagebrush at our Rocky Canyon site than at our Magic Reservoir site. We also found that cottontails consumed significantly more sagebrush than has been previously recorded. However, despite the differences between the sagebrush consumption at these field sites, we found that at each of the field sites, the proportion of leaves consumed did not vary between the study sites we looked at. So several differences that we have observed at these field sites could help explain why pygmy rabbits and cottontails consumed more at Rocky Canyon than at Magic Reservoir. One of these is the crude protein content. A previous study found that at Cedar Gulch, the sagebrush there contains more crude protein content than at Magic Reservoir, and pygmy rabbits are shown to select sagebrush plants with higher crude protein content. Monoterpene content has also shown to vary between Cedar Gulch and Magic Reservoir, with Cedar Gulch containing a greater variation of monoterpenes in the sagebrush than magic. Alternatively, food resource availability varied between the sites and could help explain why more sagebrush was eaten at Rocky Canyon. So here, these are our controlled feeding experiments. On your right side is the 50% basal diet that we offered, and on the left is the 25% basal diet offered. Oh no, that's backwards, right? Okay, sorry about that. So on the top is the average proportion of sagebrush the rabbits consumed, and on the bottom graph is the average proportion of leaves consumed. We found that, again, pygmy rabbits consumed a greater proportion of sagebrush than the cottontails on both basal diets, and pygmy rabbits also consumed a greater proportion of leaves. We found that both species tended to consume more sagebrush when they were offered less of an alternative high-quality basal diet. However, both species were shown to consume a lesser proportion of leaves when they had a less high-quality diet available to them. Studies at Sagebrush Flats, which is another study site where pygmy rabbits occur in central Washington, have shown that pygmy rabbits consume about 25% sagebrush in the summer, which is the darkest green portion on that graph, but they consume about 75 to 99% in the winter, which is still the dark green portion on the graph. Our results suggest that pygmy rabbits and cottontails may consume more leaves when they're eating less sagebrush and when more alternative high-quality diets are available, and this suggests that they may be able to dilute the toxins in the sagebrush by consuming a wider variety of vegetation. When looking at the stem diameters cropped by captive and free-ranging rabbits, we found that pygmy rabbits in both situations crop smaller diameters than the cottontails. So on the left is the captive rabbits that we measured the stem diameters for, and on the right is the free-ranging stem diameters that we measured at Cedar Gulch. We also found that free-ranging rabbits tended to crop larger diameters than their captive counterparts. Although little is known about the effects of removing leaves and stems at different diameters on sagebrush plants, it has been shown that large ungulates who take larger bites and that consume sagebrush can decrease the amount of sagebrush shrubs and the amount of concealment cover they provide. Although cottontails crop larger bites than pygmy rabbits, further studies should be done to investigate the impacts of fewer but larger diameter bites versus smaller but many diameter bites on the growth rates and biomass removal of these sagebrush species. So we found that 100% of our cottontails cropped branches at larger diameters, and then they consumed the middle of the branch, but then discarded the smaller diameter leafier apical stems, while 0% of our pygmy rabbits exhibited this behavior. These discarded apical stems, as well as fecal pellets deposited in foraging areas, may contribute to nitrogen recycling in the soil as they decompose, and may increase the growth rates of Wyoming big sagebrush and other perennial grasses that occur in the sagebrush steppe. So, in summary, pygmy rabbits consumed more sagebrush and a greater proportion of leaves than the cottontails did. Both species consumed more sagebrush but less leaves when alternate high-quality diet was less available. And cottontails cropped larger diameters but discarded the smaller diameter leafier stems to consume a lesser proportion of leaves and most likely a higher proportion of fiber. Our work exploring how these two rabbit species select sagebrush leaves and stems suggests that there is a greater overlap in winter food resources than previously thought. Pygmy rabbits and cottontails may partition their use of sagebrush differently, and therefore they may contribute to ecosystem dynamics differently in sagebrush-steppe habitats. And I would just like to thank everyone who has helped on this project, including all of these people. And I would just like to thank ASM again for this award and the opportunity to be here today. Thank you.